Okay, so last class we had finished the process of replication and so on, and also different enzymes which are involved in replication. That is what we had seen, and we had looked at what is the central dogma of the cell or of the biology and so on. So now we'll be starting with the next step and another important process, that is transcription of RNA. So again, if I go back to this process, so what we had looked at from DNA, how to make a duplicate copy of another DNA molecule through the process of replication that we had seen. Now what we are going to look at is from DNA to RNA, that is transcription process. And then the last one will be translation after this. So it's the process of formation of RNA from the DNA template, and that process is known as transcription. Now we already know that DNA contains the information with respect to the formation of specific physiological as well as morphological proteins, right, or morphologically required proteins, and it is located in the nucleus, while the protein synthesis, that is the translation process, happens in the cytoplasm. And we already know that if DNA moves outside the nucleus, it will probably get degraded. So it does not move to the site of protein synthesis, that is at the site where ribosomes are located. So that is when this intermediate step to synthesize the mRNA, and it will transfer that mRNA to the cytoplasm from nucleus. So that is the process of transcription. So transcription occurs in the nucleus during G1 and G2 phases of the cell cycle, especially during the interphase part of it, because that's when we need a lot of proteins and lot of cell organelles which are made up of proteins. So that's where more of the mRNA will be required. So obviously more of the transcription process. DNA has the promoter and terminator sites that we had seen earlier, where the transcription starts and stops. So transcription will start at the promoter region, that is before the initiation codon, and it will stop at the termination site, which is stop codon, and after that. So only one of the two DNA strand, that is 3' to 5' end, is transcribed into RNA. And this again we know, this is the template strand, or what is also called as non-coding strand or antisense strand. And from these strands, the RNA is transcribed, which ultimately what we get is 5' end to 3' end. So that is the general transcription description. Now, so this is how a transcription looks like. Again, this diagram we had seen it earlier also. This is the DNA molecule which has different nucleotide bases, and this will separate. It will cause the mRNA synthesis on 3' to 5' end. So ultimately the direction of the RNA will be 5' to 3'. So that is the same direction. And then this will get, what we'll get is a transcript, we call is RNA transcript, and that will be utilized for the processing for the translation process. So that is the general outline of the transcription process. Now, this whole of the mechanism of mRNA synthesis was discovered by this Gerard Horwitz and Samuel Weiss and Audrey Stevens. And this happened somewhere around, I guess, 1958 or so. The segment of DNA template strand which transcribed the RNA is called as transcription unit. Now remember that mRNA, what we get as a molecule, and the DNA template or DNA sequence, they are matching to each other, but not all the information, all the genetic information that is present in the DNA is translated into mRNA. Some part is being lost. That is when the exons and introns comes into picture. So that introns, they are lost over the time, like until the final mRNA transcript is synthesized. So this that is when the whole of the transcription unit is something that is the complete RNA. So as DNA is the same copied version of that is the transcription unit in the form of RNA. It contains three main regions: promoter region, structural gene, and the terminator region. So promoter region is one we can simply think of, that is where all the machinery or protein complexes will go and bind. They will start the transcription process. Structural gene is the one which actually has the main protein coding information, and the terminator gene that will say, okay, now the transcription has to stop. So let's look at this one by one. What are these different regions in the transcription? In promoter region, it is located upstream, that means before the initiation codon. Initiation codon is ATG. Before ATG, promoter region is located. And initiation codon of the structural gene, and towards the 5' end it is located. So that is 5' end. Let's say if I show you in this diagram, so let's say ATG is the first codon, that is the initiation codon. Before the ATG, that is this upper transcript that you see, which is the 5' end of this DNA strand, that is where this promoter region will be located. So this strand of blue string, that will be promoter region. And this promoter region will provide the site for binding of RNA polymerase. Now remember, we had in the DNA replication, we had DNA polymerase, which was doing the elongation of the newly forming DNA strand by polymerization of the nucleotides. Similarly, in this case of transcription, there is a requirement of RNA polymerase for the initiation of the transcription. So RNA polymerase will go and bind to the promoter region, and it will initiate the transcription. And this promoter region also helps in differentiation of the template and the coding strands of DNA. So again, you might wonder why not the other strand transcribes. So let's say, for example, this strand is transcribing for the mRNA synthesis. This upper strand will not transcribe the mRNA. And the reason being, because there will be no promoter here, rather there will be complementary to the promoter region, right? These base pairs which are on 5' to 3', they will be complementary to this particular strand. So RNA polymerase will always bind to the 3' to 5' end of the DNA strand. So that is what is the differentiation of the template and coding strands of DNA. The functional sequence in the promoter region, they are called as consequence sequences. I guess, did I miswrote this word? I don't know. Somehow I feel this is called something else. Let me just quickly check. I have a feeling that I did some gadbad. Yeah. It is not consequence, it's consensus. Consensus. I felt so. Consensus sequences. Okay, sorry about that. So the functional sequences in the promoter region, that means before ATG, whatever is present, it is called as consensus sequences. Now, what are these consensus sequences? These are the functional sequences of the nucleotides of TA. That is what. So there is only presence of adenine and thymine. So that is what is a short sequence written here. And they are called as Tata boxes. Tata boxes because they have only T and A and T and A and so on, right? And not necessarily T and A has to be alternate. They can be in any other format. So TAAT or ATT, any of that format can work. But the presence of adenine and thymine is what defines this consensus sequences. And in case of prokaryote, they are known as Pribnow box instead of Tata box. Tata box is common terminology because even in prokaryotes and eukaryotes, it has this TA sequences. But in prokaryotes specifically, it is called as Pribnow box because it's named after this guy, David Pribnow. And in eukaryote, they are called as Goldberg-Huggins box. But you can simply call it Tata box for the time being. But they have some different names based on the name of the scientist in prokaryote as well as in eukaryotes. So that is the Tata box within the promoter region. So most of the gene transcription processes, they are regulated by this Tata box. So just to let you know that in DNA, in cancer, cancer-like diseases, what usually happens is there is a particular tumor-producing protein, or what we call as tumorigenic proteins. They are more in nature, more in the cell, right? And that means their transcription and translation is happening to the larger extent. So then what is being done is the targeted cell action to the Tata box of those particular genes is done as a part of therapy. So that is when less amount of transcription of these genes, less amount of translation will also happen. So less amount of protein, and that is when tumor will be kind of suppressed enough. But again, that is not so simple as I say it, but still that can also work as one of the strategy in case of most of the protein transcription, mRNA transcription processes. Any question here in the promoter region or in the general transcription?
So
yes.
For DNA, before the DNA polymerase can come in, you also need a RNA primer, right? So this doesn't happen in transcription?
This doesn't happen in transcription because there are these Tata boxes which are kind of RNA polymerase binding site in the promoter region, so that does not really require.
Okay.
And also, like me, I'm not too sure of this fact, but we know to some extent that the first organism or first living cell was RNA. RNA was present in those cells, right? So then the evolution must have happened in a way where the RNA transcription was given as a more important process throughout the process of this evolution. And maybe that is when the DNA polymerase was not present earlier, and later on it was modified to accommodate the DNA polymerase. So that could be just one of the reasons why RNA polymerase does not need anything to transcribe the DNA strand. And why does the DNA polymerase need that RNA prime? Could be just maybe a reason. Any other question? Anyone? Okay, so then the next part is the structural gene. But before structural gene, we'll see the Terminator region. So in Terminator region, it is located downstream. Again, these are the terminologies that we usually use in the genetic study or in the central dogma studies. Upstream always refers to before, downstream always refers to afterwards. Now, before and after what? It is always contextual, but in case of DNA, it is usually referring to upstream is towards 5' and downstream is always towards 3'. So this terminator region is located downstream of the non-template or coding strand. Non-template or coding strand means DNA strand which is from 3' end to 5', right? So that is the DNA strand they are talking about. So it defines the end of transcription of structural gene. So we know what structural gene is. It's a particular code of nucleotides which is coding for a protein. That's what is a structural gene. And they at the end of this structural gene, there will be a terminator region. which has 2 or more stretches of GC. GC is guanine and cytosine pairs.
So,
Yeah.
This is the DNA strand, right?
Yeah.
Like these regions. So shouldn't it be in the non-coding strand? Because the non-coding strand will be the one that makes the the non-coding strand will be the one that's transcribed, right? Like the RNA will form on top of that.
Correct. But so like if you remember, there is always this DNA helix. That's when this diagram is being shown here. Where is it? Yeah. So let's assume this is the terminator codon. It is the terminator codon. Not, we don't have to assume it. ATG is the start codon. TA is the stop codon, right? So that is what is terminator region here. So, and when the RNA is kind of synthesized, it is also aligned with this length. So it recognizes this sequence, right? Which is ultimately a part of or complementary sequence to this. So that is what it is being said here. So it is located at the 3' end of non-template coding strand, and it ultimately means complementary to the non-coding or not non-coding. No, what myself getting confused. Where is it? Yeah. So this is the template strand. This is the coding strand. So on the coding strand, which is a non-template strand, that is where it will be present.
Yes, but the thing that's being used for coding is the template strand, right? So shouldn't the segments of DNA that we are talking about be parts of the template strand?
That they will be ultimately part of it. No, that is what I am saying. This sequence will be recognized by the RNA polymerase, which is ultimately a stop codon for it.
Yes, but the ATT part that's there, it's the terminator region in the template strand, right?
Yes.
So, like, why did they mention the other strand?
Because it is ultimately assumed, I would say.
No, I mean, what's the use of the other strand in this process? Like,
because the stop codon is on the coding strand. It is not on the what we call as the
template
strand. So that is why. And also in addition to that, there are these GC stretches. So which is GC as in, like, if the template strand has G, then the coding strand will have C. So that is the only possible option. So even along the length, it will be only GGGCC or GCGC, whatever that sequence is. So that way, these will be 2 or more stretches of this GC after this stop codon. So the moment, now you can imagine, right? Even if this stop codon does not transcribe or does not translate into any amino acid, that you know for sure, right? But as soon as after the stop codon, if there are these GC reach sequences, that will be acting as, and whether it's on this what we call template strand or whether it's on the coding strand, there it will not matter much, right? And that is when they will act as a terminating process for the transcription.
okay, so in the terminator region, the 2 strands are connected.
Not necessarily.
So, like in the diagram, it showed that.
Correct, but that they are trying to show that the translation is happening only in this, transcription is happening is only of this strand. That is the structural gene that they are trying to show. They have not really shown the, and you can see this is clearly a hand-drawn diagram. So they have not shown any GC or promoter sequence, which is promoter or terminator sequence. This is just to show. But yeah, in general, that is right. The DNA helix is not helix as such. it is kind of broken up to a certain length and until the mRNA synthesis happens. And like in a way, then you will be right. Let's say this is like, let's say these are 15 or maybe 100 base pairs after this T until this DNA helix is intact. So there are few like still quite a lot of gap, because what we usually assume is that RNA polymerase is doing this synthesis. So RNA polymerase will sit on this, it will synthesize, but along with RNA polymerase, there are so many other protein molecules, which is what that is when we call is a transcription complex. So they will need lot of space, and they keep emptying the path until the RNA polymerase walks from behind and start transcribing. So that way it is not so immediate as it is shown in the diagram. It is just a representative one. Okay, so yeah, so it has two or more stretches of GC pairs arranged as inverted repeats. Inverted repeats means they will be kind of complementary to each other, and then it is followed by a poly A tail. So that is the again, this is what we had seen in the mRNA structure as well, right? In mRNA, during the transcription process, it has a poly A tail. So that is something to simulate that situation during the transcription. This is acting as additional signal that, okay, that is the terminator region. So now the transcription process has to stop. And then this is the additional signal. And then to the mRNA, again that poly A tail is added. We will see that post modification, post transcriptional changes. So that is the terminator region. In structural gene, there is nothing more to explain apart from it is particular, it is coding for a particular protein which is needed for morphological or functional trait of a cell. This we already know. Either it is involved in the structure of the cell or it is involved in the function of a cell. That is what is the structural gene related to a particular protein. Now, there was this guy called Benzer in 1955, I guess. He used the term cistron. This we had seen earlier, which is a functional segment of the DNA. So a gene is considered as a unit of function, which is called cistron, a unit of recombination, which is called as recon, a unit of mutation, which is called as muton, and a unit of regulation, which is called as operon. So these are the different terminologies which were developed after this Benzer guy. He started studying this transcription processes and so on. So again, you just need to remember these names and what these names refer to, which is ultimately with the muton, mutation, recon, recombination, and so on. Cistron we have already studied, and operon is probably the new one, which is the unit of regulation. So there are some things called as lac operons, which is like a stretch of particular gene, which are responsible for controlling this transcription and translation processes. And that's what is this operon. In some cases, operon also refers to a stretch of 2 or more genes. That is like a kind of polycistronic, more than one gene in one stretch of functional unit of DNA. But that is a different case. So like we know, the structural genes, they can be of two types, that is polycistronic, especially in prokaryotic, and monocistronic, that is again in eukaryotic. Monocistronic because it's usually one gene is separated by the other gene with certain stretch of DNA. So that's why they are monocistronic. And we have seen this at least the this terminology called split genes, because they are not continuous sequence. There is exon, there is intron, and then there is some additional base pair sequences in the DNA structure which are not coding for anything, but they have to be present for the structural architecture of the DNA helix and the nucleosome and so on. So that is what is referring to as split genes, because it is non-continuous, or what we call as discontinuous. So that is what is split genes. And RNA splicing is another mechanism which we'll see in the transcription process itself. That splicing, probably splicing means to cut out. So DNA has your exon, intron like this. That means functional, non-functional code, functional, non-functional code. That way they have alternate exons and introns. Exons are the one which are coding for the protein. Introns are just additional units of nucleotide within a DNA. So this removal of these introns, which are not functional units of nucleotides, that is what is called as RNA splicing. So in general, the structure of the transcription process looks like this. So there will be, the structure of this coding, like whatever mRNA is synthesized will look like this. Promoter region, that is then there will be the ATG, where that's where the transcription will start. Then there is a structural gene, which is will have the what we call as stop codon, and the terminator region will be present, and then it will continue into its own stages. So that is the general structure of this. Okay, now I hope you people don't have any question in this part, because that's relatively easy, I would say. But yeah, if at all, you can ask. So the next topic, no, why did I do this? Sorry, that's a additional slide for myself. It came by mistake. So transcription in prokaryotic cells. So how does it happen? So there are several steps involved in this transcription process. So I have just listed down the points here so that we can go, like we have also seen that to some extent in the DNA replication process. Activation of ribonucleotides. In that case, in case of DNA replication, we had seen activation of deoxyribonucleotides, because they were for DNA. This is for RNA, so obviously it will be the activation of ribonucleotides, which is nothing but the addition of phosphate group and forming from that monophosphate to triphosphate. So that is the activation part. I hope you people will have an idea of that particular process. If not, you let me know. I will explain a little more in detail. But I assume because we had done this in replication, this should be okay here to just write down pointer, and then you can elaborate on that. So first step, activation of ribonucleotides, which means addition of phosphate molecule to the monophosphates so that it becomes a triphosphate molecule. That is what is activation. Then recognition of the promoter region and the binding of RNA polymerase to the promoter region. So now we already know what promoter region is, which will have the starter boxes, which will be recognized by certain proteins along with RNA polymerase. So that way RNA polymerase will be recruited at the promoter region before the ATG or the start codon of the DNA molecule. Then the exposure of the DNA bases, that is the third step, and the initiation of the transcription. Exposure of the DNA bases means all the DNA bases, they will be opened up, so that double helix which was there, it will be kind of unwound. Again, there are several other proteins involved in this. So hydrogen bonds will be broken, that DNA stretch will be elongated, and the initiation of transcription will start happening by the RNA polymerase. So this exposure of DNA bases, it means the two strand will break their hydrogen bond. The helical structure will be kind of made straight, and the transcription will start after recognizing ATG. The fourth step is base pairing, which is nothing but the RNA polymerase will take the complementary base pairs and it will keep on adding. Like again, we had seen this in the case of replication. So that is what is base pairing. Now, after base pairs are formed, conversion of ribonucleotide triphosphate to ribonucleoside monophosphate. Now, this is again to form the backbone. We only need one phosphate molecule in between the adjacent what we call ribonucleotides or adjacent nucleotides in RNA. So, for that to happen, it must convert again from triphosphate to monophosphate. So that is the step number 5. Then formation and elongation of RNA polynucleotide chain. So then the addition of those phosphate group, not the phosphate group, the sugar and phosphate backbone will be joined together throughout that mRNA nucleotide sequence, and that is what is the formation and elongation of RNA polynucleotide chain. Again, the same case like we had seen in case of DNA as well. Triphosphate becomes monophosphate, and then the neighboring nucleotides, they are joined together with each other, and then it forms a stretch of one molecule. So here in that case, similar thing happened, but just it is RNA polynucleotide chain. And at the end, obviously, when the termination sequence will be recognized, or the stop codon and then the terminator region will be recognized by this transcription assembly. It will separate, it will stop the transcription process, and then it will separate the RNA chain from the DNA molecule, because again in this case, so this RNA that will be formed up to end, this terminator sequence which is present at the end will be recognized by this transcription assembly. The assembly will fall off, the RNA molecule will separate from this DNA molecule. This is specifically called as DNA RNA complex of the transcription. So RNA will separate, and then that will come out as a RNA transcript. So where is it? Yeah, and then reformation of the DNA helix. So DNA will again fall back. The helicase, the DNA molecule will again form the double helix structure, and it will go back to its regular process. So that is the transcription in prokaryotic cell, which majorly involves these 3 main steps. That is initiation. Again, initiation refers up to this initiation of transcription. These first 3 stages, they are the initiation part. Elongation is nothing but this base pairing and the conversion of this triphosphate to monophosphate, and then formation and elongation of RNA nucleotide chain. And the termination is this last stage where the RNA is separated and the DNA again goes back to its helical structure. So these are the 3 main steps divided into several stages. So along with this RNA polymerase, it also needs certain initiation factors. It's nothing but the protein molecule. We call sometimes them as so and so factor. So there is this initiation factor, which is sigma, which is denoted by this symbol. And it is required for the initiation. So along with the RNA polymerase, this initiation factor will also go and bind to the promoter region. And for termination factor, that is rho, it is required for the termination process. So these are the 2 additional factors. Now, of course, there are quite few others, but what is there in the syllabus is these 2 in addition to RNA polymerase. Any question here? I hope this process is clear. What we what we have to do is just elaborate on these points, which I think they are okay to elaborate on. Okay, so I hope there are no questions here. So now the transcription in eukaryotic cells. So the transcription, that whatever we had seen was for prokaryote. In eukaryote also something similar happens. It just there are few additional steps apart from what we have seen in the prokaryote. So again, in case of eukaryote also, most of these things they will remain same. Activation of ribonucleotide has to happen, the binding of the promoter region, and then initiation of so the process remains same. There is no change. There might be difference in little bit of RNA polymerase from bacterial to eukaryotic, but these subtle differences will remain, but the process is remaining same, but after this processing in eukaryote there are additional steps. So that is what is being shown in this diagram, which is the promoter engagement. That means the complexes which are going to start the transcription process, they will bind to the promoter region, including RNA polymerase. They will begin RNA synthesis. They will keep elongating the RNA molecule. Now you can see here the two strands of DNA, they are shown as it is, only a little loop is being shown here. But that is not always the case, or rather this is not the case. It usually stretches up whole of the DNA molecule. Two strands are separate, like it was shown in this diagram. This one. This is how it occurs. Not necessarily the whole of the stretch of the structural gene has to be opened up and then only RNA transcription can start. Not like that at all. But quite a lot of DNA molecule has to open up depending on the size of this RNA polymerase complex that will occupy. So it's not just this small loop of the DNA which is broken with the helix structure. And then of course termination, where the termination factor will also be required, that is rho. It will recognize, along with the RNA polymerase, it will recognize the termination codon and termination region, and then it will fall off from the DNA molecule and DNA will again go back to its original helical structure. So that is the transcription in eukaryotic cell. Now apart from this, the difference, major difference in eukaryote is there are three different polymerases. Why there are three different polymerases? Because that RNA polymerase 1 will transcribe the rRNA, RNA polymerase 2 will transcribe the mRNA, and RNA polymerase 3 will transcribe the tRNA part. And then of course whatever they are called differently, like rRNA, we know there are different subunits within those ribosomes molecule. So that is what is this RNA polymerase 1 will do. mRNA, which is also called as heterogeneous nuclear RNA, because it has still those introns and exons intact. It is just a copy of DNA molecule. It is not completely processed yet. So that is what is the function of RNA polymerase 2. And 3, that is RNA polymerase 3, it will catalyze the transcription of tRNA. Again, if you remember the structure of tRNA, it has to undergo quite a lot of modification in terms of that uridine molecule, addition of methyl group or hydroxyl group and so on. So those modifications has not happened yet in terms of tRNA. RNA polymerase 3 also synthesize or transcribe 5S, that is sedimentation coefficient of rRNA. That rRNA is synthesized by RNA polymerase 3, and small nuclear RNA, that means the fragments of RNA which are smaller in size, that is also transcribed by the RNA polymerase 3. So these are the three different RNA polymerase present in the eukaryotic cell. So again, there are different types of promoters for three different polymerase, which is quite obvious. Otherwise, if the polymerase, all these three different polymerase, if they have same promoter code, which polymerase should go and bind will be always a question. So instead, its RNA polymerase will have a different promoter region, RNA polymerase 2 will have a different promoter region, and so on. So that way they will always be different promoter sequences for all these three different types of polymerases. And many additional transcription factors, they are required for binding of RNA polymerase to DNA. Like I said earlier, it's not just RNA polymerase and one or two additional protein which will start the transcription. There are so many other transcription factors which will cause the binding of RNA polymerase to DNA, and then it will allow the transcription process to happen. So that way, this process is relatively complex. Again, any question so far?
But why don't they have different RNA polymerases for prokaryotes? Because they also have ribosomes, right?
See, structural modification, they will always be there. But in this case, they are more evolved in terms of structure. In prokaryote also there are different versions, I would say. Okay.
So the RNA polymerases, they are different even in prokaryotes. But
Again,
they are not termed differently, but there will be some differences in them.
yes, but like I said, here in this case, these are completely different units altogether. There, like I will show you, if at all I find that prokaryotic RNA polymerase structure. Yeah. See now, so depending on, so this is like a complex of what we call as different proteins, and that is what is being shown here. So let's say a random example, I don't know now which subunit takes care of which type of RNA in prokaryotes, but let's assume alpha will take care of mRNA, beta will take care of tRNA, and this omega or that one will take care of rRNA. So that way different subunits can function for different RNA transcription process. So that way in eukaryotes, we already have totally different sets of protein, right? Eukaryotic DNA structure. Again, I'm not too sure. Do I see any difference here? RNA polymerase. Yeah. So means in a way, now the structure is this RNA polymerase. 1 and 3 it is showing. Yeah, means okay. Now assume that these are RNA polymerase 1, 2, 3. It says so in the diagram, but I'm not too sure of this until and unless we read the whole paper, cannot be sure. But you can see certain differences, right? You can see there is extended tail here in this polymerase 1. It's absent in this, and it is even more extended in 3. So structurally itself they are different. Yeah, so that way there is quite a lot of difference between prokaryote and eukaryote, even in terms of protein, and even this diagram talks a lot about it. Depending on what polymerase it is, it will recruit different proteins. So for example, this RNAP, I don't know, I assume it is ribonucleotide adhesive protein. RNAP. Again, I'm not sure because I haven't read this paper. So RNAP 1 is recruited by polymerase 1, RNAP 2 is recruited by polymerase 2. And depending on this presence, also the other other proteins, they will be recruited as well. So that way there are quite a lot of difference. So that is when we say no, prokaryotes are relatively much simpler, and eukaryotes, they are more evolved, and they have separate structures for separate things. Does that answer your question, Pratham?
Yes, I mean, so there is one, but it seems unlikely that one structure can do all three functions, right? Because like proteins are structure specific, right?
That is where again the difference is, because in case of prokaryote, it's polycistronic, right? So there are no introns and exons, right? While in case of eukaryote, we have introns and exons. So wherever there are introns and exons, we need mRNA to function there. Wherever there are short stretches, this is going to be tRNA, which is hardly what, 200 or no, 73 to some base pairs, right? So that is going to be tRNA. So obviously the function here is more evolved with respect to different RNA types. In those cases, that is not the case. Also, mRNA is synthesized in eukaryotic cell because it has to transfer from nucleus to cytoplasm. That is not the case with what we call in prokaryotic cell. It's everything is in cytoplasm, DNA itself in cytoplasm. So you don't need very specialized what we call machinery or proteins in prokaryote. There even nucleotides, they are simply available right then and there, so they can keep forming as soon as the transcription has happened. So that way it is much simpler system, and that is when even if we assume that there is only one RNA polymerase in prokaryote, it wouldn't be wrong, and in eukaryotic we have 3 different types, again it wouldn't be wrong, because the functions, they are drastically different. Okay, what is next? Okay, I guess we will stop here. In the next class, we will talk of this processing and post-transcriptional changes. This is the slide which explains a lot of this. You can just go over this prokaryotic and eukaryotic transcription process. So that way, this will give you some idea. We are just left with this in the transcription process. We will talk about these changes. That's where most of these things will also get more clarity. Okay, so let's stop here.